Hey, what's up everyone? Today I'm going to talk about how the virus that causes COVID-19 fuses our neurons together, giving us neurological symptoms that we see during COVID-19 virus infection. The good news is that the vaccines that have the spike protein that is shown to cause this fusion of cells doesn't have that ability, because the spike proteins are changed in such a way, they're mutated so that they don't have this ability to fuse neurons together. So I've gotten people ask me as a PhD whether these vaccines are safe, and I always had great faith in them. But this paper that I read in this past week that was published in Science Advances shows that there are potential causes for concern with vaccines. If these mutations weren't there, there would be a viral fusion causing potentially symptoms that are similar to what we see in COVID-19. This is not what I expected, because I expected the complete virus infection is what causes all of our symptoms, but potentially only pieces of it, such as the spike protein, can cause neurological effects. You probably know that COVID-19 causes neurological symptoms. When I say it that way, it may seem surprising, but you know this. It can cause a headache, can cause a fever, can cause confusion, can cause epileptic seizures, can cause a loss of taste and smell. That's the distinctive one. But it can cause more serious effects. As we all know, it's caused a huge death toll in our country and in the world. I didn't notice any long-term changes in my thinking after COVID-19. Doesn't mean there aren't any. I definitely noticed some changes while I had the infection. Let me go through some of the ways that they showed fusion of neurons in a variety of cells infected by the SARS coronavirus 2, which is the virus that causes the COVID-19 disease. COVID-19 really means coronavirus disease 2019. So when cells are infected with coronavirus, they can be lost or they die, but sometimes they become fused, and that's what we're going to talk about. Some viruses use host machinery to enter cells, and they take over the cell, and they start making their own fusogens, and these are proteins that go to the exterior surface of the cell and display itself. And these fusogens cause the fusion of cells. They decorate the outside of the infected cells, allowing fusion with the neighboring cells. And by doing this, they create syncytia. These are fused cells that have multiple nuclei and allow the virus to propagate within these syncytia. The spike protein of coronavirus must bind the human ACE2 receptor to get entry into the cell. And this is a fusogen. But this binding of the spike protein also triggers fusion of cells using this ACE2 fusogen. They showed the fusion of mouse hippocampal cells by engineering them to express different colors of GFP and then infecting them with SARS coronavirus 2 and mixing them together. They found that these cells showed the mixing of GFP in their images in a variety of cells, and that means that they're merged cells. But the mock control with no viral infection did not show any mixing of GFP. That means the GFP of one color stayed in one cell in the image, and the GFP of another color stayed in a different cell in the same image. When they mutated the spike protein that they used to infect these hippocampal cells, they didn't see this mixing of GFP. One thing that I was really excited about when reading this paper, especially since it's not a type of paper that I read all the time, because it has so much biology and cell culture work that I'm usually not exposed to, which is great, but it's nice to also hit upon something that's more familiar. And this happened when they utilized a protein called the CAID protein, which I had never heard about before. My background is biochemistry and protein structure, and so this was very interesting to me. The CAID protein is a fluorescent protein that actually has a green emission. But when you shine light on it, it shifts to a red emission. And this is because there's a chemical change that happens to the fluorophore that absorbs and emits light. So they use this capability of this CADE protein to further provide evidence that there is a fusion of cells in response to SARS coronavirus 2 infection. When they infected hippocampal cells with SARS-CoV-2, and they had also electroporated these cells, which means that they provided these cells with the genetic material for this CADE protein. It expressed that CADE protein, and it was fluorescent. When they did the infection, they then illuminated part of a cell with UV light, and so that triggered that transformation of green fluorescence to red fluorescence. And they're able to show a diffusion of the CADE protein from one cell to an adjacent neuron, which was, I think, freaking really awesome. Just provides another way to show that there is a passageway for this protein to travel to an adjacent neuron once it is infected with SARS and causes this fusion. They also looked at this fusion in a live animal called C. elegans, which is basically just a worm. It's a nematode, and this is a highly studied organism in neurobiology. They expressed fusogen P15 because it doesn't have this human ACE2 receptor, but this P15 receptor does respond to the COVID spike protein. They also expressed GFP in these cells, and they expressed this P15 and GFP into 6 mechanosensory neurons, and these are neurons that can respond to movement or touch. So this is a transgenic nematode. It has all of its natural genetic makeup, but it also has GFP and P15 in some of its neurons. When the cells were infected with SARS coronavirus 2, they observed fusion in the cells that had P15 because they saw GFP containing cells throughout the body of the nematode. And when there weren't P15 present in the nematodes, and there is a transgenic nematode that didn't have P15 and GFP only, they did not see the presence of GFP in other cells. So they use these human embryonic stem cells to show merging of the cells when exposed to the spike protein. And these cells didn't need any fusogen because they have the human ACE2 receptor that was able to facilitate fusion. They showed in different images that the fusion can occur with the soma, which is like the body of a neuron, or the neurite, which is one of the branches of a branching dendrite. They showed by labeling mitochondria that mitochondria can actually travel from one neuron to another after fusion. So these are big passageways and not just small tunnels that allow single biomolecules through. The last really interesting part I want to share with you is they showed the activity of these neurons before and after they're fused. And they showed that there's huge, significant differences between the fused neurons and unfused neurons. They used a calcium-sensitive dye to measure the activity of these neurons. And so I assume that they have an electronic trigger, and then they take images after the trigger and look for changes in the color that signal calcium has gone into the cell. And they monitored this activity over time. And so they're able to look for synchronization of neurons. They found that the fused neurons had really good synchronization or really consistent synchronization, but they had a 10% loss of activity. And some of that loss of activity is due to the merging of neurons with glial cells, which are neural cells that do not fire themselves. And so this was detrimental to the neurons to fuse with glial cells. But the non-fused neurons, they didn't have any loss of activity, and they had a variable range of synchronization. So sometimes they were synchronized and sometimes they weren't synchronized. So there's a big difference there. I'm guessing that the synchronization, the perfect synchronization of the fused neurons is not optimal because we need differences in our neurons to have unique transmission and more complex transmission. Because if your neurons are firing all at the same time, there's no complexity or there's no real information there. You want a complexity of information. I'm not a neurobiologist, but I did do a few episodes on the chemistry of memory. I'll put the link for those videos below. Super interesting stuff. So I know a little bit about it, and I'm very interested in neurobiology. All right, that's all the content I have for today. I hope you enjoyed this episode. Hit subscribe if you want to learn more about biochemistry. I'm going to put on a real broad array of topics on this channel that I think are interesting, and I hope you do too. All right, take care. Bye.